So I want to thank everyone for coming out to tonight's Deep Talks event. My name is Mike Ray. I'm the Media Relations Manager here at the Sanford Underground Research Facility, and just so excited to be here for this great night celebrating science and science communication and science outreach. I want to thank you all for coming out. It's great to see such a nice turnout for this event. It's great that it's not 30 degrees below zero outside, right? That's a very good thing. But we also need to thank some of the sponsors who have really made this event possible. The sponsors for the free beer, you can't go wrong with that. That's a good deal. And the sponsors that make this event happen. And to do that, I've got Michelle Kane. She's the director of the Surf Foundation. She can speak more to our sponsors.
Awesome. Thank you so much, Mike. How many of you, this is your first Deep Talks? Oh, I love it. We hope that you have the opportunity to connect with others, discover some new ideas, and above all, leave excited about science. That's what Deep Talks is about. As Mike mentioned, I am the director of the Surf Foundation, and we are the nonprofit arm of Surf. We support public outreach, like tonight's Deep Talks, STEM education, teacher professional development, cultural programming, and building intellectual community through the new Institute for Underground Science at Surf. So tonight's sponsors are Chris Mitchell from Edward Jones and Spearfish, Chuck and Jolene Linkton-Wallner. Yay, you can clap for them. They are steadfast supporters of Deep Talk since 2019. And their donation plays a critical role in what we're doing tonight. And that is nurturing a passion for science and education, which benefits our local community and our global audience through our live and recorded events. A special nod tonight to our friends at Crow Peak Brewing. And since 2015, they have been quenching our thirst here at Deep Talk. So when you're in Spearfish next, please be sure to visit them and to thank them. So to all of you who are joining us tonight and to our listeners who are tuning in during the recording, we invite you to become a friend of Surf and help with the supporting that helps us bring you this great education and science tonight and our other outreach efforts. Donations can be made at the donation box on your way out or online at sanfordlab.org/foundation. So thank you for your support of Surf, and I'm going to turn it back to Mike, and enjoy your beer.
Thank you so much, Michelle, and thanks again to those sponsors. They're the reason we're here. We are starting tonight with a special awards event. The Sanford Underground Research Facility hosts the CORES Award, which stands for the Communication and Outreach Experiences in Science. And this year's recipient is the South Dakota Mines Museum of Geology. I want to thank them for being here. And those with the Museum of Geology can come up if you want to. We'll be handing out the award here in just a minute. The CORES Award is given annually by SURF to an individual, a group, or a business or organization that demonstrates an ongoing commitment to public outreach efforts across the states of South Dakota. The award recognizes those who undertake STEM communication, specifically, those who are champions of science, those who communicate about science, and for over a century, the Museum of Geology has exemplified everything that the CORES Award stands for. They have been doing multiple free public outreach events throughout the years, along with STEM advocacy. They have an expertise in things like paleontology, mineralogy, the geology of the Black Hills, and their outreach keeps so many people interested in STEM and engaged in STEM. I think paleontology has been called the gateway to STEM, as any young person can tell you. They know fossils better than we do many times, so they're great. The award this year is an engraved piece of steel that was part of the Nobel Prize-winning neutrino experiment that was undertaken by Ray Davis here at SURF. And the base for the award is actually a piece of wood that came out of the Yates shaft as part of the recent refurbishment of the Yates shaft. This is a 90-year-old piece of wood that was a part of homestake history and a part of surf history today. I also want to introduce Mike Headley. He's the Executive Director of the South Dakota Science and Technology Authority and the Laboratory Director at SURF. He's going to join us here to present the award to Dr. Darren Paniak, the Director of the Museum of Geology, and Kaylee Johnson, the Assistant Director of the Museum of Geology. And we're going to do a little photo op here. So maybe we can do the gather around the award with a handshake sort of thing. Stephen, our photographer, is going to help take this photograph if there's any in the audience who want to take photographs too. Thank you for this. We'll keep the halo out of there. That's great. Thank you. Let's give another congratulations to the Museum of Geology. I'm going to turn it over to Mike Headley here.
All right. Thank you so much. Congratulations. We're really happy to have you here and honor the work that you're doing here in South Dakota. So thank you so much. Thank you. All right. So my Fermilab colleagues promptly got up and left the room when I was going to recognize them. Are they coming back in? Okay. So I think if you live in the area, you know we've been excavating big caverns underground for a big neutrino experiment called the Deep Underground Neutrino Experiment. And the third cavern has been something that has been in progress until just recently. The first two caverns have been completed for a bit. Actually, the first cavern that was completed well a number of months ago in 2023. And the first big detector cavern was completed the first week of December of last year. And then actually this week, the last large blast in the second big detector cavern was completed, was it today? Today. And so the breaking of rock is largely completed for the Long Baseline Neutrino Facility Deep Underground Neutrino Experiment. And apparently my FRA colleagues are not going to come in here. So, but anyhow, congratulations to Fermilab and to all their subcontractors and to our team as well, and partnering with them to help them get the job done. Huge, huge milestone for everybody working here on the site and everybody here in the area who's, a fan and supportive of SURF. So, thank you. So the excavation contractor, Tyson Mining, today, they must have gamed this. That they hit today 1 million hours of work without an injury. So really amazing safety record. So, all right. Thank you, Mike.
And thank you, Mike. The Dune experiment, of course, is going to be pioneering physics in the world. It's a wonderful thing. But besides amazing world-class physics, we're also doing amazing world-class biology. And that's what Dr. Tanvi Goval is here to speak about tonight. Her pioneering work in microbiology and biological and chemical engineering at the South Dakota School of Mines has led to several different breakthroughs. One of those recent breakthroughs that Dr. Goval and her multidisciplinary team of researchers made included isolating some microbes from surf and other areas that can sequester carbon dioxide. This is very exciting and potentially revolutionary work. And I've got to get Dr. Goval set up with a microphone. Let me just I'll hand you this if that's all right. And we'll go from there. She can speak to this, of course, much better than I can, so I'm going to be quiet now.
Okay, thank you, Mike, and thank you all for having me here. While having all the chats and all the talks outside, I literally forgot I have a talk. So I'm trying to bring back my memory now, what I'm here for. I have just joined as an assistant professor in Chemical and Biological Engineering Department in School of Mines. I joined in August 2023, so it's like less than a year for me. But I have been associated with School of Mines since 7 years now. I did my PhD, then postdoctoral fellowship, and now an assistant professor. What I do, I am a biologist, and I belong to a huge group of biologists at School of Mines. As a background, I'm a microbiologist in that category of biologists. So we keep on looking for those tiny microorganisms around. So tiny microscopic bacteria that you cannot see with your naked eyes, but are around everywhere. Maybe just, above your head right now, lot many microorganisms. So what I do, we keep on hunting them, keep on mining them. Wherever I go, my eyes are more looking for, there can be bacteria, this can be a spot for bacteria. We bring back the samples. I do a lot of study on their DNA. Then I study their genetics. What can they do? What cannot they do? If needed, we change their DNA, basically change their genetic stuff. We do collaborate with a lot of computer scientists, AIML scientists. Okay. And then finally, my main objective is how can I use that microbes for purposes that are relevant for the environment? Either do remediations, treat something, or maybe make them worth enough for industries. That's what, as a biologist, we had been doing. One of my, this is my PhD work that we did. It's just an example of what we do with microorganisms. Like one of my PhD work was we had to find a bacteria, we find microorganisms from the nature that can break the agricultural waste, corn stover, sugarcane waste, any waste. No chemicals, no physics, nothing involved. The microorganisms eat that agri waste and they are making bioplastics out of it. So it's like purely biodegradable plastics, degradable in 30-day time period. That was one of my first encounter with microorganisms world. I would say the polymer we are making, it's having a melting temperature, degradation temperature of 420 degrees Celsius. That's one of the hardest or the most thermostable polymer that we have found. So this was one of my first encounter with microorganisms. And since then I have, been doing a lot with microorganisms. Starting with pre-treatments, I break plastics now, I break agriculture waste now. We make lot of biopolymers, we make lot of biochemicals coming from microorganisms. I really like to do genetic engineering of the microorganisms. We are slowly starting to work with microorganisms to make fertilizers. Like how can you use microorganisms and fertilizer so that you don't have to provide chemical fertilizers. So a lot of different projects and we can keep on talking about it. But one thing as a microbiologist, biologist, chemical engineer, biological engineer, what we have to do is mining. We have to keep on looking for idle places where we can find a good amount of microorganisms. Our favorite spots had been, we work a lot on thermophiles, means microorganisms that live at temperatures of more than 50 degrees Celsius. And one of our favorite spots had been earlier, like going for rapid city landfill compost sites. We keep on looking for hot springs around in the world. Yellowstone has been one of our favorite sites where we find a lot of hyperthermophilic microorganisms that can even live under the boiling point temperatures of the water. Then we keep on working a lot with even the opposite set of microorganisms, psychrophiles. Psychrophiles grow at cold degree. They grow below 10 degrees Celsius. So we have been receiving through our collaboration samples from Antarctica, Arctica. Then another set of category of microorganism we have been very interested in the past is piezophiles, means bacteria, microbes that can tolerate a very high pressure. So for example, 10 megapascal means 10 times or maybe 100 times atmospheric pressure. So we go deep into, we don't go, but we have connections. So we get samples from deep floor ocean mines, etc. And during these excavations, one of the thing was surf has really given us a lot of microbes. Surf, I joined School of Mines in 2017. Since then, I have been coming almost each year to surf for finding samples. But before that, my mines group, Dr. Professor Sani is also here. He was probably the first person, his group, who went deep under the mine in 2006. Since then, lot of undergrad students, graduate students, we have been coming back and forth to the mines, collecting the samples. We have collected the samples from probably all the levels of surf, starting from 1700, 4100, 4850 is one of our favorite spot. And lot of just, as I said, I was telling somebody when I go to the mines, I don't look what is in the mines. I keep on looking where is the bacteria, what sample I can collect. When we leave the mines after our trips, we are full of so many samples that, literally, it's like by the time we reach the lab, we forget what are the samples and from where do we collected it. Students are like so busy collecting their tubes and filling up their tubes. Literally by the end of the whole trip, we literally forget what have we what. But As of now, if I would say, in our culture collection in School of Mines, we have close to 300 unique microorganisms, and Surf Mine has contributed out of them close to 80. So my 30% of the microorganism we have in the laboratory is coming from the Surf Mine. I do have in my collections a lot of thermophiles, psychrophiles, piezophiles, bacteria. So these three categories in particular. One of now surf has not only given us a lot of microorganisms, but one of the first project that my professor he got was a bug remedy project, an NSF funded project. Was This project was aimed towards finding microorganisms in the nature or studying how the methane gets regulated in nature. So what are the cycles that methane gets oxidized for? So methane is a potent greenhouse gas, you all know that. It's 20 times more dangerous than carbon dioxide gas. So this project was funded by NSF. The title or the aim was finding in the deep mines, deep earth environments, and surf was perfect spot for them. And trying to see how can we remediate the methane. Can we find from the nature some real solutions that can help us eat the methane gas? And this project, I would say, did provided them more than 100 unique extremophiles in the surf core at 4100 levels. They were able to find some microorganisms. There are a lot of lists, lot of namings here, but I would say the peculiarity in the surf mine, we were able to find some bacteria that eat methane gas as their food. Their genomes were previously unknown. So they were having all this unique category of genetics that made them very eligible for eating the methane gas. This project also was able to support workforce of more than 100 people, lot of undergraduate students, lot of graduate students, lot of publication records we have recently from this project going on. And our project on methane was one of the biggest project that really inspired us to go next. So next, we thought one greenhouse gas is methane. We already found the good microorganisms from the surf plant itself that can eat the methane, remediate the methane. What about carbon dioxide? Do we have the environment in surf that can help us, take away the extra carbon dioxide we have in the environment? And that's when NSF funded for us another project. This project is being done in collaboration of School of Mines with some industries people, but geologists from School of Mines, civil engineers, environmental engineers, biologists, we all have been associated with this project. Why are we studying carbon dioxide? Carbon dioxide is a greenhouse gas again. And since it's first time, especially I would say just in the last decade, there has been 50% increase in the levels of carbon dioxide in the environment. Now, what is the solutions if we have to reduce the levels of carbon dioxide? Everybody says just reduce the level of emissions, correct? That's what we all believe. Now, as a biologist, we believe that if the people, if the community, or if the world cannot stop emitting carbon dioxide, can we do something to trap them? So that's where this project landed. Now, in terms of, this is just a very background. Plants, the green plants, they are the biggest sites for capturing carbon dioxide. They take all the carbon dioxide, they do the photosynthesis, they trap the carbon dioxide. But the plants, when they decompose, when they die, when they complete their life cycle, they emit all the carbon dioxide back to the atmosphere. Second option you can trap the carbon dioxide is you take the carbon dioxide, pressurize it, convert it from gas phase to liquid phase, supercritical nature, and just inject them very deep inside the earth, more than 2,500 kilometers down, using some oil and gas reservoirs that are depleted. You can use coal mines, you can use deep saline aquifers where there is brine water. These spots have been invested for more than decades now. For years and centuries, that scientists are trying to trap the carbon dioxide in these locations. What is the advantage? Maybe it's a huge spot. It can actually trap more than 80% of the carbon dioxide which is available in the environment. But what is the disadvantage? It's a costly process, and more or less, it's a very destable process. there are high chances at any point of time all the CO2 will come out back into the atmosphere. So this is a very viable process, probably one of the most explored, but it has its own disadvantages. So next, what is the other options we have? So in the nature, water is another reservoir that keeps on trapping a lot of CO2. There are tiny microorganisms in the ocean. They also do photosynthesis. They eat the carbon dioxide. They die. They get settled down at the bottom of the ocean floor. Or the carbon dioxide gas, it can react with water. It can form carbonic acid. It's like whole little chemistry that goes on. But whenever that acid comes in contact with some minerals like calcium, magnesium, iron, they form carbonates. And what is a carbonate? It is a form of a rock. So you can convert carbon dioxide in the atmosphere, store them as the rocks. Now that rock formation or mineralization process can also happen on the surface of the earth. That's we call mineralization. Same process. If you see, carbon dioxide need to react with water, form some kind of acid, then the acid forms react with available minerals and they get trapped in the form of rocks. This is second most exponential growing method of trapping the carbon dioxide in the atmosphere. You can do them ex situ, means find some mining sites which have exposed rocks, or we can even do them in the laboratories. So what we have to do, we have to find the correct piece of rock that can provide iron, magnesium, calcium, required mineralogy, and then nature does the work. One thing, this is the most explored method, kind of considered the most safest method of trapping carbon dioxide gas, but it's a very slow process. This process, if it happens at room temperature, normal temperature, pressure, it takes more than 10 years. Even I would say like 10 years is very slow. It would take hundreds of years to just this, I don't know the main geology, but it takes hundreds of years to capture the carbon dioxide in the form of rocks. Now, our group's challenge was, can we accelerate this process? Can we accelerate the process so that something happening in example 10 years can happen in 1 year? So that was the challenge that came to us as part of this NSF project. And what we found was, in the nature, there are some tiny microorganisms. They make an enzyme. So what they do, they secrete some enzymes outside their body. So it's all sticking outside in their body. Their all body is covered with this enzyme. This enzyme has the capability to accelerate the whole process of mineralization by more than 10%, like I would say 10 times. So they allow you to form the carbon dioxide reaction with water. The first step is so much accelerated that if you can explore these microorganisms, you have the chance of trapping CO2 as mineral in a period of, let's say, 1 year. And that was the target we gave to ourselves. Can we accelerate the process of mineralization from maybe 10 years to 1 year period? And what we found was that if we have to accelerate the process, we have to find microorganisms in nature. One problem, at room temperature, normal pressure, this process is very, very slow. If we have to accelerate the process chemically, we have to either go at a very high temperature. High temperature means at least close to 100 degrees Celsius, or we have to go at a very high pressure. High pressure would mean close to 500 bars or more than 500 times the normal atmospheric pressure. And the question that came in front of us as a biologist was, can you find in nature that one bacteria that can grow at 100 degree, that can tolerate 500 times atmospheric pressure? Our answer was yes, we can find. And you know where we came? We came to surf mine. 4850 level, and this was a trip that happened in 2021. There was some drillings being done under as a geothermal project by DOE, and they were drilling some 4100 levels, some deep cores of the rocks in the surf mine at 4100 level. So we got very interested in, and we literally asked them. We were one day, the whole day we spent at the surf mine, and we were like asking them, can you please give us that rock sample? Just little bit of that rock sample. And we took that rock sample with us back to the laboratories. We crushed them, grinded them. We did some experiments with all the 4100 level collected rock samples that were drilled up till like 200 feet horizontal drillings. What we found? That will come, but one of the undergrad students, while we were doing those drilling, that was one of the funniest pictures. She also find she she was doing something. We were looking into the rocks and she was looking at the ground, and we found that she found a pot of something. We still don't know what's there, but there was something coming out from the underground or the floor of the mines in 4100 level, and that was smelling like sulfur. Lot of sulfur, lot of probably acid. So she got so much interested in not the rocks but collecting that sulfur sample, and she did a very like good work in finding the microorganism. She found a lot of acidophiles here, acid living bacteria, microorganisms. But more or less, this is one of our funniest story that we always share, that she was not interested in rocks anymore. She was looking at those spots everywhere then. But let's go ahead. What we found? Did we found life? So people ask us. You were at 4100 level. They were drilling at the rocks, which might have been very ancient rocks. Correct? Did you find something there? Yes. We were able to find not many microorganisms, but successfully we found at least 4 of them. They are, if you know, if I just can quickly give on some introductions on the naming. Agrobacterium, frankly, it's an agriculture bacteria. Agrobacterium is one of the microorganisms that nature has in agricultural soils that helps the plants improve the nitrogen fixation rates, improve the fertilizer rate. What was it doing in surf? We don't know. Then we really don't know. So all the microorganisms that are listed here, they are not known. They were not supposed to be in the surf mine. They were not supposed to be knowing to exist inside the rocks. What were they exactly doing there? We had to literally ask our undergrad student, did you do the culturing well? I mean, are they like not the contaminants of the lab? Are they the contaminants of the surf? And 2-3 different groups of people, 2-3 different groups of undergraduate students actually confirm the same story. So don't ask us what are they doing here, but they are doing something or the else. We used some microscopic experiments then. We were looking them for our carbon dioxide project basically, and we did some microscopic experiments, and we used a very powerful microscopy that we have now in School of Mine, which is a nano IR instrument, and we could locate that on their surface of the microorganism, they were actually making carbonates. So we have the evidences of these bacteria having the enzymes, having the required chemistry, having the required biology to trap the carbon dioxide and form the rocks. And this is the next step of our project was we started to use these microorganisms in the lab. These are our very tiny reactor. This is the level of experimentation we are doing at this point of time. But we had small, tiny test tubes. We filled them with some carbon dioxide dissolved in water as carbonic acid. We put in these microorganisms that we found at the surf mine. We put in some extra pieces of rocks so that it can just give them some space to live. We put them under some conditions, different conditions of temperature, pressure, pH. We were doing a lot of experimentations. And we found that these bacteria are actually surviving 500 bars, means 500 times the atmospheric pressure. They are surviving 80 degrees Celsius as the temperature simultaneously. We did those experiments for 10 days of period. Frankly, our instrument died after 10 days. It was not able to sustain the instrumentation, was not able to sustain the pressure continuously of 500 bars. But even after 10 days, these microorganisms were alive. So a lot of experimentation is still again happening, still on these experiments. And we are not showing the results, but we have also increased the temperatures or pressures now to 800 bars. That's the maximum limit our instruments are allowing us at this point of time. Instruments are dying, not the bacteria. That's the beauty we have got from surf. I really don't know if we would have even gone to Yellowstone National Park. We would have gone these unique microorganisms set. Maybe those microorganisms would have been unique on temperature side, but surf gave us a combination of bacteria unique at temperature and pH. Now, at this point of time, what we realized was that the samples or the rocks we collected from the mine, we were able to grow 4 bacteria in the labs, but we found the evidence of fossil DNA in the rocks of maybe the microorganism that had been living. They were present some point of time, and a big chunk of undergraduates now working in the laboratories, they are doing some genomics at this point of time. So they are trying to find the DNA inside the rocks. What is the DNA going to tell us? Probably maybe not microorganism living today, but something that left or they were living maybe decades ago. This experimentation frankly has been going on for 2 years now. The DNA that we extract from these rocks is so tiny that students are not able to do anything, but the students are not ready to give up. Every time the batch change, students come, new students, they do the DNA isolation, but we are very hopeful that we are very close to finding what are the category of microorganisms that surf had been hiding. So this is our current progress with the project. More or less, I'm not showing here, but at surf, we also find a lot of microorganisms that can breaking plastics for us now. Again, the question mark was surf having a lot of plastics initially? Why are they hosting the microorganism that can break the plastics? We really don't know. But surf has given us as a biologist lot of microorganisms, lot of different applications. One successful project completed after 6 years, finding new novel microorganism that can eat methane as a gas. Second group of experimentation just getting completed now, where microorganisms can actually, probably I won't say eat, but they can trap carbon dioxide from the environment in the form of rocks. So yes, so surf mine I think is incredible if you want to explore more in terms of stopping the climate change. All the greenhouse gases I think can be assimilated by a lot of surf microorganisms. So this was the one of the presentation or just a small research that we have been doing at School of Mines and we wanted to share with you. Thank you so much. So I will be open to any questions if you have.
Thanks for coming out again. I want to give anyone a chance in the audience who would like to ask Dr. Goebel a few questions for this great presentation. I can follow around, come to you with this microphone and hold it. So anybody have any questions for Dr. Goebel this evening on her work?
So ask me questions on biology, okay?
I think you may have actually already answered it, but I think I missed it. Once you've isolated an organism, how do you determine that it has the characteristic of being able to capture carbon dioxide? Is it just the presence of the enzymes you mentioned, or is it a bigger process?
It's a very good question. So we did a lot of processing on them. One thing we start with is, okay, so if you have to find what a microbe can do, you can do 2 routes predominantly. You can study their genetics, means study their genomes, what it is made up of. So that gave us a pretty good hint initially that these microorganisms are having the required enzymes that can help us fix the carbon dioxide. So they had a category of enzyme which is we called as carbonic anhydrase that accelerate the reaction of carbon dioxide with water. So we found this microorganism had these enzymes. And then as a second step was doing the experimentations.
Okay, thank you.
Anyone else? Hands up, anyone? Yeah.
Very much enjoyed the talk. For the 4100 sampling, you were able to harvest fresh rock, fresh core, but you mentioned you were able to find some non-living, I don't know, ancestors or other evidence of microorganisms. Mm-hmm. Is there an opportunity to further explore non-fresh core, looking for evidence of these types of microorganisms?
Oh, I mean, we would be happy to. We keep, as I said, our main target is find the samples. Give us all the rocks. We would be very, very happy to find them. Just anything. So yes, that's correct. One, and that was one of our intention, that if we can go get other rock samples which are as old as they can go, it sometimes becomes very difficult because the DNA there is in form of fossils. So it sometimes becomes very difficult to find even a tiny point of DNA inside them. Sometimes we can see that there is DNA, but that DNA maybe is not good enough for us to do further experimentations. But yes, if you have a lot of rocks lying around, we can come back.
Anyone else? Anyone else? Other questions?
On the microorganisms that you're finding in the rock samples and underground, is there a way to determine how old they are? Are they always growing down there, or have they been trapped? And when you open them up, then they come to life? Or has anybody determined that yet?
Maybe, I mean, there are technologies available, there are techniques available where you can answer that. So the people who work on the fossils, they have all the instrumentations, experimentation skills that they can tell you what period of time they were living from. That's what I'm trying to say, that in the rocks, we found a lot of microorganisms that maybe they are dead at this point of time. We can just see that they have the evidences of DNA, but they did not came back to life as soon as we moved them. It was just 4 of them for us. We were successful in finding 4 of them who revived, jumped back to life. Now, if I go back a little bit, so not all, but these microorganisms are known as, we call it a category of gram positive. They have a very thick outer skin, very, very thick. They make a lot of spores, which we call endospores. So they are like, they create like an eggshell around them. Maybe that shells protected them. That's what we were asking ourselves. What are they doing here? We might have been exploring, finding, trying. We did not expected life there. When we went there, we were excited to have the rocks, but at least we did not expect it. So maybe that hard shell course enabled them to live.
Others? Anyone else? I want to ask one question if I can. The fossil DNA that you talk about, could that give you insight into genetic engineering future microbes that would maybe sequester carbon better? Could you use the fossil DNA to inform the genetic engineering?
Mike always asks us so good questions that we sometimes run out of answers for you guys. And he has been a source of inspiration and, ideas. Maybe that is the next project idea we can throw to NSF.
I think we have a few paleontologists in the room who might know more than I do, that's for sure.
Yep.
Any more questions from anyone in the audience? But before we all go, we want to offer Dr. Goval an award for taking her time here to speak, and that is a framed photo of the Sanford Underground Research Facility. And this frame was also made from Yates Shaft timbers by our own Stephen Kenny, the photographer tonight. So I'm going to hand this over to Dr. Goval. And Stephen, may we even want a photo back there with you. So thank you so much for coming out, Dr. Goval. And thank you all for coming out. We want to thank again the list of sponsors that Michelle was so great to list. Our next Deep Talks, I believe, is on February 8th. And that is going to be a talk with the Native American Studies program at Black Hills State. We're looking very forward to hearing from them. It's going to be wonderful, and we'll have more out on our website about that event. So please come back on February 8th. And if you have any follow-up questions, don't hesitate to reach out to any of us at SURF, and we can connect you with Dr. Goval. I think we need to get you some more rocks and some more biology. We got to keep this work going.
Anytime, even if you have something around your houses lying here and there, just call us. Yep.
Thank you all for coming out. Have a wonderful evening.